So these are essential amino acid. Mnemonic is his mat will fly, his mat will fly. Histidine and arginine are semi-essential, semi-essential. Methionine, I told you, methionine is having sulfur. Sulfur means methionine is essential, cysteine is non-essential. Threonine, largest, O try 3 sari, threonine essential, tyrosine and serine non-essential. Tryptophan, bulky side chain, essential. Live, essential to live. Branch chain amino acid, essential. Phenylalanine is essential, yes. Tyrosine is non-essential. Lysine, lysine, yes, lysine is basic amino acid. Basic amino acid are basic need in our body. Clear? Yes, transporter of ammonia in muscle is alanine. In, yes, urea cycle we can, yes, discuss urea cycle, but before that let me tell you these questions. Okay, this is this, suppose this structure is given, they have given this diagram. This is cathode, this is anode, and they have given this, and yes, 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 aspartate glutamate negative charge, arginine histidine lysine positive charge. Okay, this is, this is, this is, this is. If I ask you to sequence these, you will say this is negative charged, can anode is positive charged, and negative charge. Yes, yes, this is arginine. Yes, this is, this should be arginine maximum positive charge, that arginine, this should be glutamate. Yes, negative charge. No, yes, this is histidine positive charge, this is glycine. I can keep them in this sequence. So arginine maximum positive charge, maximum, yes, maximum towards cathode. Glutamate negative charge, maximum towards anode. Histidine, and this is glycine. Okay, we can we say due to transamination hepatic encephalopathy occur? Yes, you can't say like that, because transamination is different. This is not transamination, this is joining of ammonia to alpha ketoglutarate. These are transamination. Okay, these are transamination. Okay, so this is about the structure. I know that you are little bit confused about the structure, so I skip this topic for the time being. Yes, and yes, you have read this already. Yes, figlu is a metabolite of, figlu is a metabolite of histidine. Figlu is a metabolite of histidine. Histidine. Histidine is converted to figlu. Figlu is form amino glutamate. Form amino glutamate. Okay, oh sorry, form amino figlu. Yes, figlu is converted to glutamate. Glutamate, yes, glutamate. Yes, in this tetrahydrofolate, yes, histidine is converted to Figlu, form ammonium glutamate. In this, Figlu is converted to glutamate. In this reaction, tetrahydrofolate is converted to form amino tetrahydrofolate. Form amino tetrahydrofolate Figlu is converted to glutamate by, in this tetrahydrofolate is converted to form amino tetrahydrofolate. Now we found that, so in this question, Figlu is a metabolite of histidine. Figlu is a metabolite of histidine. Okay. Yes. And yes, if folic acid is deficient, then Figlu will not be converted to glutamate. Figlu will not be converted to glutamate. And Figlu, if you give loading dose of histidine, if folic acid is deficient and you give loading dose of histidine, this histidine will be converted to Figlu but will not be converted to glutamate. Figlu will be excreted in urine. Figlu will be excreted in urine. We say Figlu test positive. Figlu test positive. So question is asked, Figlu is a metabolite of histidine and Figlu test is done for. Figlu test is done for, then the answer would be folic acid deficiency. Folic acid deficiency or folic acid. Okay, clear? So this is phenylketonuria is congenital amino acid metabolic disorder, enzyme deficient is. Please do reply. Please do reply. Phenylalanine hydroxylase. Well, if I take a different color, if in the same question, if I say rare variant, this question is asked in PGI frequently, rare variant, yes, then the answer would become dihydrobiopterin reductase. Very good. This is recently asked question. Most accepted method of HbA1c estimation is ion exchange chromatography. What is chromatography? Yes, this is frequently asked question. What is chromatography? Yes. Yes, what is chromatography? Suppose I give you a mixture of two protein. Suppose one is protein A, which is small. Protein B, which is large. So I gave you mixture of two protein. One A is small, B is large, okay, and I ask you to separate these two protein. I gave you the mixture of these two protein. A is small, B is large. I want to separate these two protein. Then I did gel filtration chromatography. What is the name of this technique? Gel filtration chromatography. Don't worry, I will tell you very nicely. Graphy. Yes, whenever you will find the word chromatography, you will say there are two phases. One is mobile phase, second is stationary phase. Mobile phase, which is moving, stationary phase, which is not moving. This solution, which is made up of two types of protein, this is mobile phase. I took a column in this column I am modifying the technique a little bit, but concept is correct. I took this column cylindrical, just like this bottle. If I cut from this and this, okay, this is cylindrical. Okay, I took this column and I attached gel molecule. I attached gel molecule here on the wall. To simplify, I put it on the wall. I attach gel molecule on the wall. What is gel molecule? This gel molecule, just like this is a channel, like means in this there is a gap. This is gel molecule, in this there is a gap. In this gap, yes, we took this gel in such a way, this gel in such a way, in this gel this small protein can enter but large protein can't enter. You can easily assess that in this gel small protein will enter, large protein can't enter the gel. Means gel molecules are having, yes, gel molecules are having, yes, pore inside it, and we pass the solution from this. When we pass the solution from this, large protein, protein B has only one option to go straight, because protein B is large enough, it can't enter the gel. But small protein A has two options. Few molecules will enter the pore, few molecules will go straight. Few molecules will enter the pore, few molecules will go straight. So in the first solution comes out, that is elute. This is called elute. The first solution which will come out will contain mainly large protein or small protein. Yes, this was mixture of protein A and B. A were small, B were large. When we pass a solution, B were have only all or one option. All the protein B protein will go straight, but few molecules of A protein will enter the gel and will take time. So which protein will come out first? Which protein will come out first? Which protein will come out first? Which protein come out first? Large protein. And this is called gel filtration chromatography. Means gel filtration chromatography. Yes, you have given the correct answer. Gel filtration chromatography. In the first solution, mainly, yes, large amount of B and very small amounts, few amounts of small molecule A would be there. Means this is not completely purified. What I said, this is not completely purified. Few molecules of few molecules of small and majority of large protein will come out. Now what we will do, we will pass this solution again to second column. We'll pass this solution again to the second column. Again it is having, yes, gel, having gel, yes, gel, gel. Okay, yes, having gel. Again, yes, small molecule will enter this and large will come out. Means again and again we are doing this. Every time we will get more and more purified protein. So this is not a chromatography, is not a simple process that we will put and everything will be separated immediately. It would take time, and it is not a Sieve-like structure. Remember when this question was asked, gel filtration chromatography, which protein come out first? Student thought, when we do filtration, if I take a sieve and will pass the solution of large protein and small protein, who will which? Yes, if I take a sieve, if I put the mixture of two proteins, large protein will remain in the sieve and small protein would come out. Yes or no? So, if I ask you ultrafiltration, what I said? If I ask you ultrafiltration, ultrafiltration or dialysis, ultrafiltration, which will come out first? Please do reply. Ultrafiltration, which will come out first? Yes, yes, this is the fact. This question, when this question was asked, at that time student thought, yes, I described in such a way you could catch the answer, but in exam when this question would be asked, you will not think about the mechanism, you will not think about the mechanism. Immediately you will think filtration. Okay, this is filtration. We take a sieve, we will pass the solution, small protein will filtrate, large protein will remain the surface. No, this is not a simple filtration, this is not ultrafiltration, this is not dialysis, this is gel filtration chromatography, which will come out. Yes, gel filtration chromatography, which protein come out first? Large protein. I don't know, Ravi, you are giving the previous answer, but yes, you know, remember, you understand the concept. Okay, in gel filtration chromatography, large protein come out first. In ultrafiltration, simple filtration, dialysis, small protein, yes, small protein come out first. Got the point? Yes, ultrafiltration, dialysis. So this is different. Yes, ultrafiltration or filtration is same for you. Means filtration we are, yes, simple. Yes, yes. Ultrafiltration or dialysis is different thing. Just like you take a semi-permeable membrane and a small molecule will pass through that membrane. Yes, so just to, yes, yes, even if you forget this, rarely question asked, but this question was very important question at that time. In gel filtration chromatography, it is not simple filtration, it is chromatography. Large protein will come out first. Clear? And chromatography is not a single step, it is multiple step done again and again, and every time you will find more and more purified form, means if you do 10 times, purification is increased. If you do 100 times, purification is increased. And many times you will find that in this test we can detect the thyroid level, thyroxine concentration up to this unit. Means you can do chromatography up to this time. Why we have limit? Because you are passing this solution. Whenever you pass this solution from one column, some solution is stick to the wall and gel. If you do 100, 10,000, 1,000, 10,000, all the solution is attached to the wall, column and nothing in your hand. That's why we have limit to perform these cycles. Okay, we can do this cycle up to a limit. That's why we have limit of purification of a particular protein by chromatography, because we can't perform this chromatography multiple cycles. Yes, otherwise in last you will find nothing, because all the solution will be attached with the column. Okay, got the point? Have you understood the concept of chromatography? One is mobile phase, static phase, mobile phase, static phase or stationary phase. Yes. Now ion exchange chromatography. Ion exchange chromatography. What is ion exchange chromatography? If I give you a mixture of two protein A and protein B. A is positively charged, B is negatively charged. I want to separate. I take a I take a column. I take a column that this is, but let me complete this. Okay, ion exchange chromatography. I take a column. Yes, in this I attach CMC, carboxy methyl cellulose, carboxy. I told you. No. Let me complete this topic. Let me complete the topic. Yes, ion exchange chromatography. In ion exchange chromatography, I attached carboxymethyl cellulose on the wall of column. To simplify, I am saying wall of the column. Otherwise, we attach in the network in the column. But you forget about this. Yes, carboxymethyl cellulose is having which charge? Can you tell me? Carboxymethyl cellulose. Can you tell me? Yes, which charge? Carboxy. I told you, wherever carboxy group is there, negative charge, negative charge, negative charge, means we will pass this solution. This A, yes, very good. A would have positive charge, it will interact with CMC. A will bind to CMC, A will bind to CMC, and B will come out. We will purify. So you purified. Yes, you provide protein on the basis of their charge. So ion exchange chromatography purified on the basis of charge. Yes, gel filtration chromatography, protein is purified on the basis of size. And now affinity chromatography. Affinity chromatography. Affinity chromatography. What happens that yes, affinity chromatography, suppose this is a solution. This is a solution of two protein A and protein B. Suppose protein B is antigenic. Yes, you know proteins are having antigenic property. Yes, suppose I gave you a mixture of two protein and protein B is antigenic. What you said? Okay, this protein B is antigenic. I will take a column. I will attach antibody at the wall of column. When this antigen will go, this antigen will bind to antibody, because antigen has affinity for antibody. Antigen has affinity for antibody, and protein A will come out first. So affinity chromatography, antigen can be purified by affinity chromatography. Suppose a protein is yes, I take another scenario. Yes, I know that page is full, but yes, if you have little bit space, try to write. Suppose there is enzyme, there is one protein is enzyme. Yes, protein B. You all know all enzymes are protein. So you found that there is a mixture of two protein. Out of these two protein, one is enzyme. You want to purify these two protein. What you did? You attach coenzyme here, coenzyme here. Okay, and when you pass the solution, enzyme interact with coenzyme, and the other protein will come out. Okay, because enzyme and coenzyme are friend. When they will meet, they will talk for some time. They will talk for some time. After when they will talk, meanwhile the protein will come out. Suppose this A protein is enzyme, we attach coenzyme, and you know enzyme and coenzyme have affinity for each other, means they are close friend. They met after so long time, after MBBS 3-4 years. Definitely they will spend some good time, enjoy. Meanwhile B will go. But this affinity, but this ion exchange chromatography, they are husband wife, they will tightly attached, they will not leave. Now they are committed. Okay, in this ion exchange chromatography sometimes it becomes problematic. Then with this A protein remain attached to CMC. Yes, tightly attached, because husband wife, okay, can't be separated easily. Okay, so then many times we take legal help and we cut with the help of other compound which take these A protein. So that is different thing, no need to discuss at this platform, and it's not required at your level. Okay, simply this is ion exchange chromatography, gel filtration chromatography, affinity chromatography. Enzyme antigen can be purified with the help of this affinity chromatography. What is high performance liquid chromatography? Last topic, high performance liquid chromatography. Yes, high performance liquid chromatography or high pressure liquid chromatography. High performance or high pressure liquid chromatography. Yes, both are same. Yes, sometimes HPLC P4. Yes, HPLC P4 pressure, P4 performance. Yes, we all know that performance is increased by pressure. Yes, we could not read biochemistry in whole 1 year, but with the help of this pressure of PG entrance, now we are studying. Yes, we are studying in 4 or 5 days, because we have pressure, our performance is increased. So we are increasing the performance, we are increasing the performance by increasing the pressure. Yes, enzyme, coenzyme, antigen, antibody, both are type of affinity chromatography. You can apply pressure on gel filtration chromatography, you can apply pressure on ion exchange chromatography, you can apply pressure on affinity chromatography. These two are example of affinity chromatography. Yes, so you can improve, means what happens, I told you, suppose you pass this solution through this column, it take 1 minute to pass the solution. Means if this traveling take 1 minute, means 1 minute one cycle, and I told you multiple cycles are done. If I increases the pressure in 1 minute, if I increase pressure, in which when we will increase pressure in 1 minute, suppose 100 cycles. So if 100 cycles in 1 minute, performance will increase. So you are increasing the performance by increasing the pressure, means in the same time multiple cycle can be done. So you can apply pressure on gel filtration chromatography, you can apply pressure on ion exchange chromatography, you can apply pressure on affinity chromatography. So high pressure, high performance liquid chromatography may be based on size, charge or affinity. Size, charge or affinity. Okay, so this is a high performance liquid chromatography. Yes, I have given you idea. Yes, and yes, this is gel filtration chromatography based on size. Okay, dear friends, so we have completed this chromatography